All right, these are your notes for DNA replication for AP Biology. So we learned the structure of DNA, and now we're going to get into how does DNA actually replicate. Remember, DNA replicates in interphase during the S portion of interphase, which is DNA synthesis. So we need DNA to replicate precisely so that each daughter cell will receive equal amounts of DNA, and that they're identical. So once they figured out the structure of DNA through Watson and Crick, there were a lot of different researchers and scientists trying to figure out how does DNA replicate itself. And so one of the research groups were Messelson and Stahl, and this was back in the 50s. They had actually 3 hypotheses of how DNA replicates. So their first hypothesis was that DNA replication is semi-conservative. And so what that means is that one old strand is kept with each of the new molecules. So the new molecules contain one old strand and one new strand. You can kind of see it in the picture where the parental DNA is both dark blue. And then after the DNA is replicated, each of the strands has a dark blue strand, which is the old strand, and then you have the light blue strand, which is the new strand. And so this hypothesis was called semi-conservative. Hypothesis number 2 is that DNA replication is conservative, that the double strand, the original strand, maintains intact, and then the new DNA is completely new strands on both sides. You can see that in the picture where the parental DNA is both dark blue, and then after it replicates, you have the original strand is the same, and the new strand is completely new, with both of the strands being that light blue color. And hypothesis 3 is that DNA replication is dispersive. So that means, well, maybe the DNA, like when it copies, it kind of gets cut up a little bit, and you have pieces of the old and the new kind of intertwined and interspersed in both of the new strands. And so you can see that in the picture, that it goes dark blue, dark blue, light blue, and maybe it's just a mix. And so that is called dispersive replication. And so so these scientists had 3 hypotheses. So which hypothesis was right? They figured out through research and testing that the semi-conservative method is correct. And that is the one where you have an old strand kept intact on both of the new molecules, and the other side is the newly formed DNA. So that each strand acts as a template for the other. And so you can see in the picture here that if you have a mutation, it will carry through the new generations of DNA. Okay, so how does replication start? It starts by a replication complex, binding at the origin of replication. So if you go to your diagram, the origin of replication is this sequence, origin, I'm going to change my color, replication. It's just basically a sequence on the DNA that it is recognized. It's actually recognized by a RNA primer. And so if you look on this picture, you can see this little orange thing right there. That is RNA primer. And so there's going to be one on both strands of DNA that it will be copied. So RNA primer is there, and RNA primer is right there as well. So label that. Okay, so they recognize this base sequence. And so once they recognize it, that kind of starts a new enzyme called helicase. Okay, helicase. Helicase is going to unwind that DNA. So helicase is represented by this brown triangle. Okay, that brown triangle right there is helicase, and basically what it's doing is just unwinding the DNA, or unzipping the genes, they call it. Okay, and so as it's unwinding the DNA obviously is going to want to wind back to a double helix shape, but it doesn't because there are helix destabilizing proteins, and those are the green boxes here. They're going to help hold it open. All right, so as the DNA helicase unwinds the DNA, we are forming a Y-shaped region. Okay, I'll show that in blue right here. See the Y? That is a Y-shaped region, and that is called the replication fork. Okay, so that is the replication fork right there. And so otherwise, this big yellow ball we haven't defined yet, but that is another enzyme on the next screen called DNA polymerase, and that's the one that's actually going to make our new DNA. So this box here is also pointing to the DNA polymerase. It's poly, M-E-R-A-S-E. So replication is proceeding on 2 strands. And that is because you need both strands, because it's semi-conservative replication, we have to do both strands with new DNA. So nucleotides are always going to be added to the 3' end. So if you remember, DNA has a 3' end and a 5' end. So notice the 3' ends are right there. So that means that if we're starting at the 3' end, we're going to move toward the 5' end. So one of the strands is going to move this way, and the other strand will be replicating that way. And this is all being done by DNA polymerase, which is the yellow circles here. So let's label that on our diagram. DNA polymerase. Okay, so that is that yellow circle. All right, so the DNA strands are elongated in opposite directions because of the way the DNA molecule is with the 3', 5'. They are being elongated in opposite directions. So we have 2 different kinds of strands. We have the leading strand. Okay, the leading strand is going toward the fork. You can see that direction of the arrow. And the lagging strand, which is right here, the lagging strand is going away from the fork. So you can see that with the arrows there. Okay, elongation is going to go smoothly on the leading strand. You can see it's going to keep going, and as the DNA gets unwound, that leading strand will continuously just keep going and replicating. However, on the lagging strand, it's going to go, I'll use blue, it's going to go this way, and then as the DNA unwinds more, it's going to have to back up and do more. So that's why it's called lagging, because we're going to have little holes there. So we'll talk about that in a minute. Let's label the rest of our picture. So remember, this area right here is our replication fork. Replication fork. And then this is just the DNA helix. Okay. Okay, so we have leading and lagging strands. Let's label these again. This one is the leading strand that is going toward the fork. Both of the strands are going toward the the replication is happening toward the 5' end. So the leading strand is going toward the fork, and the lagging strand is going away from the fork. And you can see right here, if we started right here with our RNA primer, we're going this way. It's going to have to back up and start over here. So we have these holes that happen on the lagging strands, and we're going to have to fill them up. So those holes are called Okazaki fragments. Okay, and this box is just the RNA primer. So it says, as the fork grows, both new strands are elongating further and further and further. The lagging strand fragments, these fragments that are here, those Okazaki fragments, are actually 100 bases, so like 100 letters to 2,000 letters long. That's how bad they are. So obviously we have a problem with the lagging strand that we're going to have to fix in a minute. So the lagging strand is growing in a discontinuous manner because of the size of these Okazaki fragments that results. And so that's why they call it lagging strand. So in order to fit it, we're going to need another enzyme to seal those fragments up. So it says, notice that the lagging strand is always growing away from the replication fork. The gaps between the Okazaki fragments are joined together by DNA ligase, and so that is represented on your diagram by the purple circle. And it's kind of in white here on my diagram, but it's hard to see. So DNA ligase is what's going to seal those fragments and that strand all up, so we have a perfect strand. Where the leading strand does not need to do that because it's going toward the fork, we don't get any gaps in the sequence. So leading strand. So just listing all the enzymes together, the helicase is the one that unwinds the DNA. The primase, which I didn't talk about, but that's the one that puts down the RNA primer that initiates the whole replication process. Then you have DNA polymerase that is adding bases to the strands. seals the Okazaki fragments up, it got cut off on my outline here, is ligase. Sometimes they call it DNA ligase. And then the one that's actually going to wind the molecule back up when we're all done is actually called DNA gyrase. So obviously mistakes can happen. So once the DNA replication is complete, DNA polymerase is going to proofread its work and go back and fix whatever mistakes there were. And so if there's a wrong letter, so like if T is not bonded to A, they will fix it. All right, the rest of these notes is review on histones. So we talked before about how big DNA is and how does it actually fit into the nucleus. And so it fits in the nucleus by these things called histones. Remember, they look like the yo-yos, and I'll show you a picture in a minute. But how it works is that histones are positively charged proteins, and that positive charge is coming from the R group on their amino acid. And so if you think about DNA, DNA has phosphate groups, which is negative, all those negative oxygens. So the negative and the positive attract, and that's why DNA wraps around these histones. When DNA and the histones are together, and they are called nucleosomes. So nucleosomes are basically 8 histones. So here's the histones, the yo-yo. If you get 8 of those, that is what's forming a nucleosome. So nucleosomes are 8 histones with DNA wrapped around it. They are part of the chromatin, and its job is to prevent the DNA strands from becoming tangled up. All right, a little bit of side information on cellular aging. It says the replication process never entirely completes at the very ends of the chromosomes. They're never like finished. But DNA is protected at the ends of the chromosomes because at the very ends of the chromosomes there is no true genetic information that matters. And so that kind of blank information, the ends of the DNA, are called telomeres. And so what happens is as we age, those telomeres become shorter. They are repaired and lengthened with an enzyme called telomerase. But as you get older, that enzyme telomerase gets lost and doesn't function as well. And so that's what causes you to age. Or they think that's a cause of aging. Research is still being done on this topic.